an inspiring faculty member. Our first speaker is a young and inspiring faculty member of our department, Assistant Professor Ko Ming-ju, also President's Assistant Professor, received his first BSc Honours degree in Chemistry and Biological Chemistry from NTU in 2012, and both PhD and postdoc in Organic Chemistry from Boston College in 2018. Dr. Ko was one of the visionaries in the MIT Technology Review Innovators Under 35 Asia Pacific 2021. His other achievements include Asian Core Program Lectureship Award, China, Thailand in 2019, NUS Inauguration Grant, NUS 2019, President's Assistant Professorship, NUS 2018, Bristol Myers Squibb Graduate Fellowship in Synthetic Organic Chemistry from 2016 to 2017, La Martina Family Graduate Fellowship in Chemical Synthesis in 2016. His research mainly focuses on the sustainable catalysis, and since 2018, he has established a program on sustainable TM catalysis at NUS under three areas of research. One, ion catalysis, nickel catalysis, and single atom catalysis. He has also contributed his expertise to our students by teaching the core chemistry modules such as CM3221, organic synthesis, and CM4228, catalysis. Ladies and gentlemen, get ready for a virtual journey towards base metal catalysis for sustainable chemical synthesis. To kick off the event, please allow me to welcome Prof. Ko to take us along the catalysis. Welcome, Prof. Ko.
All right, thank you very much for the very nice introduction. So good morning, everyone. I hope everyone is getting ready to start the day. So today I'll be talking more about catalysis, and you might have heard of the term before, and how we make use of catalysis to access chemicals in a more sustainable fashion. So to give more background on myself, I'm Singaporean who graduated from Victoria School and Victoria Junior College before I moved to NTU to do my undergraduate studies, and then pursued PhD studies overseas from 2012 to 2018. So I spent about 5-6 years overseas before I came back in 2018 to begin my independent career at NUS. So always welcome to visit us. So my research in our group, we mainly focus on organic chemistry. And so I understand that not everyone is familiar with this area. So I'm going to talk about what is organic chemistry in the first place. So essentially, it is a fundamental study of compounds, basically carbon-containing compounds, understanding the properties, the reactivities, how they behave in the presence of different reagents and different conditions. And once we know all this knowledge, we can use this information to maybe design new reactions to access a wider set of compounds, or to meet the demands of a human society by producing certain chemicals, and also to meet other challenges. And why is organic chemistry important? Because it is part of our daily life. Right? It occurs from in various types of categories of compounds, including pharmaceuticals, food, explosives, paints, OLED displays, cosmetics, and so on. So understanding how organic chemistry works would be akin to understanding how these compounds are actually generated in the first place. But besides knowing that, it also involves understanding ourselves. Why do I say that? Because organic chemistry is actually part of life. If you actually consider like all living things actually are made of certain building blocks, right? For example, proteins, lipids, carbohydrates, DNA, and all other genetic materials. And if you look closely at the molecular level, you will understand that all these macromolecules are essentially made up by simple building blocks. And these simple building blocks are organic molecules, amino acids, fatty acids, monosaccharides, nucleic bases. they all contain carbon. So they're all classified as organic molecules. And so understanding organic chemistry would be also, it can actually branch out to other areas of chemistry, including biochemistry. Understanding how these molecules function at the molecular level, and also how these actually bring about life. So it is actually important to, if you want to better understand how living things work and how we work in our own body. So what can you do with organic chemistry other than just understanding how things work and knowing how things are created? Yeah, at NUS, I actually teach a course that involves learning new reactions. That means how chemicals react with each other. And we learn and we make use of these new reactions to construct molecules. For example, it is akin to assembling a jigsaw puzzle, right? A jigsaw puzzle where you have, or assembling a Lego model where you make use of all these Lego building blocks and assemble a more complex system. So these building blocks are akin to your individual organic molecules, and you just assemble them through a series of chemical reactions to construct a more complex system. And that more complex system has various functions, right? Can be a drug, can be a polymer used for material science, and so on. So understanding organic chemistry will allow you to branch off to other diverse fields which have various applications. So there was one famous chemist known as Casey Nicola who once said that organic synthesis is an art and science of replicating molecules that occur in nature. But I would argue that it goes beyond just creating known molecules, but it also involves making unknown or unnatural synthetic compounds, which have multiple functions. One example that probably strikes more into our lives is developing a drug for COVID-19, for example. If you actually read the news a few months ago, it was mentioned in Straits Times that there is an antiviral drug that is essentially a small molecule drug. Small molecule meaning it's an organic molecule with low molecular weight. And this drug, for example, is called molnupiravir. And this is actually essentially synthesized in the laboratory by a series of chemical reactions, starting from basic building blocks and through a sequence of steps, you get to this molecule, and can potentially be used as a drug to treat COVID-19. And this is just one example of a small molecule drug. You can go and search online for yourself how many known drugs are essentially made out of small molecule that can be assembled by organic chemistry. So it also involves not just replicating or making molecules that are known in nature, but that also allows you to synthesize new analogs that are unnatural, but potentially with very tremendous, important function. So, and those who want to know in the context of Singapore, so these are some statistics, those who like statistics. In 2015, for example, it has been estimated that if you look at the overall GDP of Singapore economy, chemical synthesis and manufacturing constitutes a large part of the economy. It's a key pillar, employing thousands of people. So there are thousands of people in the chemical industry. And when you talk about chemicals, it ranges from small chemicals to macromolecules, including material science. So it is an important part of Singapore's economy, and it's still growing, even though there's this pandemic, but it's recovering, and it will always be one of the most important industries for the survival of Singapore. And besides chemical synthesis, I also research in catalysis. So those who are not very familiar with what is a catalyst, is a catalyst is a molecule that accelerates a chemical reaction without being consumed. That means it will be recycled all the time. And if you might be familiar with this curve, this curve where you have a starting materials XY going to Z, going through an activation energy barrier. A catalyst is like a substance that provides an alternative pathway that's drawn in red that goes through a lower energy pathway. And that means that we just need to consume less energy to get from starting materials to products. So figuratively, it's like climbing a mountain. It takes a lot of efforts to just climb the mountain from the bottom to the top. So a catalyst is like a tunnel. It provides a channel for you to get from one side of the mountain to the other side using less energy. And obviously getting the job done faster. So that is the function of a catalyst. And catalysis is probably one of the most important things that has ever been created in human society. And it occurs in various aspects. It is used in various industries to produce chemicals. It is also used as one of the important ingredients towards the drive for sustainability, green chemistry. You use catalysis to potentially reduce the impact of global warming and trying to reduce the impact of climate change and so on. So there are many aspects of catalysis. It's a course that I actually teach at NUS when you reach year 4, if you actually choose to join NUS in the future. So if you combine organic chemistry and organic synthesis with catalysis, that will lead to amazing breakthroughs. Over the years, the last 20 years, there has been a number of Nobel Prizes awarded to this area. I just show a few, and you could just, if you have the time, you can just log on and see for yourself what are these chemists, who are these chemists, and what are they awarded for. Essentially, they're all awarded by developing catalyst systems that allows you to accelerate reactions to make important molecules for various functions. And these are some examples shown over here from 2001 to, sorry, this is 2005 to 2010, and the latest one that just won a Nobel Prize this year, if you actually are aware, is the latest Nobel Prize in chemistry involves the development of organocatalysis, essentially using small molecules as catalyst systems to accelerate reactions to create important molecules. So now back to what we actually do in our lab. We essentially work on catalysis, obviously, and at NUS we have a number of students who are essentially interested in solving problems and challenges that we find in organic synthesis. This is really an overview of what we do. But essentially, we are involved in developing catalyst systems derived from base metals. Base metals are essentially non-precious metals compared to precious metals, right? Non-precious metals, for example, iron and nickel, they are abundant, they are cheap. And because they're abundant and cheap, that reduces the cost overall for making a molecule. But in addition to that, it allows us to, if you look at the periodic table, you will know for yourself there's this D block elements that allows us that you can potentially make catalysts from. So these base metals derived catalysts also exhibit unique properties that have different reactivities, allows us to assemble molecules in exciting ways and often shorter ways. If you can make a molecule in a shorter way with a base metal catalyst, you will potentially reduce the energy and resources that you need because each step constitutes waste. And that's why we can also minimize waste. So that is part of the drive towards sustainable chemical synthesis. So some examples that we have done over the years. I'm not going to go into the detail because this might be very unfamiliar to most of you, but just imagine that you can just sprinkle this powder, which is a catalyst, very cheap catalyst, sprinkle some of that into the mixture of organic molecules, and you can synthesize new products like magic. And these new products can be used for various applications, including preparation of drugs and other building blocks for a number of synthetic developments. Some examples are this. That's another example where we use a nickel catalyst that looks, if you draw the X-ray crystal structure, this looks like a robot. These are more small molecular machines, robots that can catalyze or accelerate reactions. Reactions that are useful, that can be applied in the context of complex chemical preparation. These are all examples of alkenes found in various biological active molecules and drugs that we have managed to make in our own lab by taking advantage of catalysis. So this is just a more complex version of another reaction that we managed to accomplish. Again, the purpose is to make libraries of drug molecules in a more efficient manner so that we can minimize the impact on the environment. At the same time, still access these drug molecules for different applications. So we also we also use catalysis to merge molecules together to assemble carbohydrates, which you might have heard of before. And we can even conjugate carbohydrates to peptide molecules to create glycopeptides, which have various applications in biochemistry and also in drug development. So overall, the entire gist of what we do is essentially creating catalyst systems that are cheap and readily accessible, and then use these catalysts to merge molecules together to make complex chemicals, which have a number of applications. And what we do is just one aspect towards sustainability. There are a number of aspects in this area, and a number of our colleagues in our department actually engage in this field of chemistry, and I'll encourage you guys to go onto our website and then look and see for yourself what are the diverse areas that different professors do in their groups, and how they make use of chemistry to tackle the challenges that we face in the human society. And I think that's all I have for today. Thank you very much.
Gentlemen, let's move on to our next speaker in this event, Assistant Professor Zhao Ziqing Winston. Dr. Zhao received his double BS degree with honors from the California Institute of Technology and his PhD from Harvard University. His research focuses on the development and application of advanced imaging-based approaches to quantitatively probe the biophysical chemistry of chromatin dynamics at the single cell level. Dr. Zhao has received many honors for his accomplishments, including the NUS Presidential Young Professorship and the Young Individual Research Grant Award from the National Medical Research Council. At NUS, he also teaches undergrad modules in physical and biochemistry, such as biomolecules and spectroscopy and imaging in biophysical chemistry. Ladies and gentlemen, please allow me to welcome Dr. Zhao to tell us about shedding light on the inner life of a cell with chemistry. Welcome, Prof. Zhao.
All right, thank you for the kind introduction. Can you guys hear me and see my slides?
Yes, we can hear you.
You should see some molecule blinking on and off in the middle of the slide. If you see that, then my slide is working.
Yes, you can see
great.
your slides.
Okay, thank you. I must first apologize for my little coarse voice because I'm having a cold. So in case anybody couldn't hear me clearly, please just type into the chat box. As mentioned, I'm a biophysical chemist, and today I'm very happy to share with you some of the exciting science at the interface of chemistry, biology, and physics, and in particular, how we can use some of the concepts and tools from chemistry and physics to illuminate the biological processes that takes place inside a living cell, such as the one shown here. So first, a bit about myself. I did my secondary schooling in Singapore, and then I went to the US to study for my undergraduate studies at Caltech. And then I went to Harvard to do my PhD in biophysics. And in 2015, I came back to Singapore and did my postdoctoral training with A*STAR, and before joining NUS about 2 years ago. And my main affiliation is with the chemistry department, although I'm also affiliated with this thing called the Center of Bioimaging Sciences, where most of the bioimaging related research in NUS is being done. It's part of the Faculty of Science and endeavor. So as a primer to what I'm going to talk to you later, I would like to show you guys two very short videos or animations that I shamelessly stole from YouTube. And each video depicts a very important biological process that takes place inside the cell. And let's see if anybody can recognize what's going on in them. So the first one is actually quite long, but I'm only going to show you a very short clip of it. And you guys can feel free to watch the entire thing by yourself. Okay, I'm going to play. Okay, that's a clip I'm going to play. So you guys should see there is a molecule that looks like a human being with 2 legs, and he's walking along a linear track and dragging a very big ball at the back. So anybody can want to guess what is this thing that is shown in the video? It's part of oh, someone says kinesing. That's absolutely correct. Yeah. That's wow. It's a great first answer. In fact, when I show this to my students at NUS, many of them don't know what this is. But I think Ethan got it correct. This is kinesing. And for those of you who don't know what kinesing is or didn't have exposure to biology before, don't worry. Kinesing is a kind of protein molecule that belongs to a class of molecule known as molecular motors. And molecular motors are basically nanoscale machines that carry out the process of molecular transport inside the living cell, such as the one shown here. And in this case, kinesing really looks like a human and it steps with 2 legs, right? And exactly how we will step. And also, but in contrast to what most of us can do, this kinesing molecule can drag a much bigger ball, which is called cargo, than what we normally can do, right? So you can consider a human being dragging a big trailer at the back. So that's quite impressive. So the second video, just turn off the sound because otherwise it will give you what it is. So what you see in the second video is a linear helical shaped molecule. Then we will see various smaller molecules, ball-shaped molecules in red and blue, hopping on and off from time to time. And now this blue molecule is going to hop on and rotate along this helical molecule and then move for some distance before it, wait for it, okay, it's going to rotate for a bit more time and then it's going to do some magic. Ding, you see here, it changes conformation and it's going to do some magic afterwards. So anybody want to guess what on earth is this? It's another important process takes place inside the cell. So what is this helical molecule? This long helical molecule. That shouldn't be too difficult to guess. DNA, yes. Okay, so then what is this blue and red molecules that move in a helical fashion on this DNA molecule? That's a bit difficult. It's okay, never mind. I'll tell you. These ball-shaped molecules are called transcription factors, and they are also extremely important protein molecules that are involved in the process called gene expression. And gene expression is a process by which the information encoded in the DNA gets converted into protein. And of course, without gene expression, all of us will be dead. There's no life without gene expression. So again, so basically, that's so yeah. Oops, sorry. Let me just get rid of this laser pointer thing. So as I mentioned, the first animation shows kinesin working on microtubules, which is important for intracellular transport. And the second movie shows the binding and the binding dynamics of transcription factors on DNA, which is important for gene expression. So all this seems very cool, right? But you might be wondering, what does any of this have to do with chemistry at all? Well, the answer actually is a lot, because our ability to observe and measure the biological processes inside the cell is fundamentally rooted in chemistry. So in some sense, the living cell is really the new test tube for biochemists of the 21st century, as very aptly summarized by the title of this perspective article published in Science magazine some years ago. So what I'm going to tell you in the next 15 minutes or so is 3 examples of how chemistry can actually play such a key role in this endeavor. Okay. The first example, how can we visualize all those processes as depicted in those animations? Of course, there are certain elements of artists' imagination in those animations, but a large part of it is actually a faithful representation of what really happens inside the cell. And to do that, we actually need to visualize these kind of processes. So how do we do that? Well, I think a very straightforward answer is that we can use light, right? If a molecule we want to see can emit light, then we can see them, right? It's a simple set. But how can we make them emit light? Well, there are actually many ways in which a molecule can emit light. But by far the most popular way for doing that in a living system is by fluorescence. Okay. So fluorescence is actually a very common phenomenon around us. For example, the neon lights you see on streets outside restaurants actually emit fluorescence. And for those of you who watch Big Bang Theory, I don't know how many of you do, but I'm a big fan of Big Bang Theory. And if you do, you will remember in one of the episodes, there was this kind of luminous fish that Sheldon was very fond of. He put inside his bedroom, right? And this luminous fish can actually glow in the dark, like this. And the mechanism by which this luminous fish glow is also fluorescence. So fluorescence is everywhere around us. It's from a physical chemistry point of view. Well, from a physical chemistry point of view, fluorescence is actually the emission of light due to electronic transitions. Okay, you guys might have learned in your previous classes about electronic transitions in transition metals, which is why they're often colored, but this is the same kind of electronic transitions we're talking about here. Okay, so in terms of energy levels, we can draw a very simplified scheme like this, in which the electronic ground state is called the S0, and when we shine light on a molecule, the molecule can absorb a photon of light of a certain energy, which is going to excite one of the electrons to a higher energy state, which we can call S1. And then within each state, there are multiple vibrational levels, which are these fine lines here. So the excited electron can first decay to the vibrational ground state of S1 via a process called vibrational relaxation, which dissipates part of the energy it absorbed as heat, but not as light. But that's not all, because the molecule will ultimately have to fall back down to the ground state. And it is that process that it can emit excess energy as light. And this light is called fluorescence. So as a result of these two electronic transitions, there are usually two spectra associated with each type of fluorescent molecule. And spectra basically is a plot of the intensity of light as a function of wavelengths. as you can see here. So there are two spectra here, one in blue, which corresponds to the excitation process, and the other in red, which corresponds to the fluorescence emission process. So if you take a look at you, one thing that you immediately see is that the excitation wavelength is usually shorter than the emission wavelength. In other words, the emitted photon has less energy than the absorbed photon, which of course is simply due to the loss of energy during the vibrational relaxation process. Okay, so this is the basic fundamental principle about fluorescence, which forms about 90% of the visualization done in living systems. But all this is great, but there's another problem, which is that most of the molecules inside the cell actually are not fluorescent, or are most weakly fluorescent. So they're not bright enough for us to see. So in order for us to make use of fluorescence for visualization, we actually need to somehow label those dark biomolecules with something that can actually fluoresce. And these molecules that can actually fluoresce are called fluorophores. And again, there are many, many types of fluorophores. They can be organic, inorganic, as well as protein-based biological fluorophores. For example, some of the classic fluorophores I'm showing here include fluorescein, benzo, and rhodamine. And rhodamine refers to a huge family of fluorescent dyes, which continue to be used today. Then we also have some more recently developed dyes, for example, called cyanine dyes, and probably the most widely used fluorescent dyes for bioimaging today is this group of dyes called Alexa dyes. Okay. And these new dyes are usually brighter, and they are more photostable, and also structurally more diverse. And also they cover a much wider range of colors, from blue all the way to near infrared. So pretty much the entire visible spectrum we can see with our eyes. There's a dye somewhere in the spectrum that we can use. But I think despite the structural diversity, if you stare closely at these structures, one common feature that you will see among many of these fluorescent dyes is that they usually have aromatic groups and as well as double bonds that are connected together via single bonds. So together they make something called a conjugated system. For example, this really long system here. And by conjugated system, what we mean is that their pi orbitals actually line up with each other, and the pi electrons are sort of delocalized, so to speak, freely to move within this conjugated system, which of course makes the electronic transitions that we talked about earlier easier to achieve. So these are the organic fluorophores. We also have inorganic ones, and the best example of which is something called the quantum dots, which are actually semiconductor nanoparticles, which consisting of a core usually made of cadmium or selenium, and as well as an outer shell, which made of usually zinc sulfide or something like that. And these quantum dots are actually extremely bright and also extremely stable, which means you can shine light on them for hours and they're not going to go dark. Okay, and they also have the unique property that their color can be tuned by changing their size. So for example, you see that slightly bigger quantum dots show up as red, but smaller ones show up as blue. So that's a very useful property. You can just tune their color based on size, right? And finally, which I think is probably most interesting, are the biological fluorophores, which of course protein-based. And the best example of that is something called the GFP, which stands for green fluorescent protein. I think some of you might have heard of this before because it completely revolutionized how biological systems can be studied. And this achievement was actually very deservingly awarded Nobel Prize in Chemistry back in 2008 to these three scientists. So this GFP revolution originated from this very unassuming looking jellyfish called Acuria victoria, from which this GFP protein was first extracted. If you take a closer look at the structure of the GFP, which is shown on the right, it actually looks like a barrel with sort of 11 strands shown in yellow here, surrounding something in purple, which is in the center. So the strands on the outside is called beta strands, and the purple helix in the center is called the alpha helix, which I think some of you might know are the basic structural elements of proteins. Okay. And in terms of photophysical properties, GFP have excitation maximum in the blue to cyan range and emits fluorescence in the green range, and hence it's called green fluorescent protein. It refers to the wavelengths in which it emits. Okay. So then you might ask, so how does a protein emit fluorescence, right? Because this is not something that we usually would associate with protein. Protein can do all sorts of things, but emitting light is not one of them. So if we were actually to strip away the beta strands on the outside and expose the chemical moiety in the center, which is shown in ball and stick here, that is a group, that small group that is actually responsible for giving out fluorescence in GFP. And if we zoom in this further, we can see the molecular environment around this moiety, which is basically this thing that I'm trying to outline with my laser pointer. This thing in the center is the so-called the moiety that gives out fluorescence. And you can see that it is in contact in various kind of chemical interactions with the surrounding environment within this protein molecule. So this is an extremely important feature that is true for almost all fluorescent proteins. That is, the function, the stability, as well as the properties of fluorescent proteins are highly dependent on the molecular environment within the protein. And in fact, until this molecular environment is achieved, the protein is not fluorescent. Okay, so then you may ask, how is this process achieved? So it turns out that this process involves a sequential intramolecular reaction that converts 3 amino acids in the GFP, namely serine 65, tyrosine 66, and glycine 67. It first undergo a cyclization reaction to generate this orange cyclic structure, and which then removes a water molecule to form this CN double bond here via dehydration reaction. And finally, it undergoes a deprotonation reaction to form from this phenyl group to form this anionic moiety, which is fluorescent. That's the one shown in green. Sorry, I hear some. Is that a question? Oh, sorry, never mind. So as you can see, this process is quite complicated, but all of this can actually takes place inside the GFP by itself without even needing any catalyst from the jellyfish. So basically, this whole process is autocatalytic, which is quite amazing. So that's just GFP. But if we only have one kind of fluorescent protein, that's going to be very boring, right? Because it's like taking a black and white photograph. And nowadays, of course, we know that color photograph is a norm. So we need to have multiple colors. And indeed, this is possible because there's actually an entire palette of fluorescent proteins that have been developed by chemists over the past 30 years through extensive molecular engineering. And you can see they span almost the entire visible spectrum, from the blue all the way to red. And more interestingly, many of them are named after fruits, which bears the same color. For example, you see tomato, orange, strawberry, cherry, grapes, and plum, and as you have it, right? That's pretty cool. And when you combine these colors, then we can use this palette to do some amazing things with biological samples. For example, in this very famous painting drawn using bacteria that can express different fluorescent proteins. So you'll see that I think there are probably 6 colors here, and each of course correspond to one fluorescent protein. And there are situations where even 6 is not enough. So what people thought is, okay, perhaps we can express different levels of different fluorescent proteins in different cells in a combinatorial fashion. And this led to something that is even more remarkable called rainbow. So if you take this word apart, rainbow basically is a combination of brain and rainbow, right? So basically, what it says is that we can achieve a sort of a rainbow depiction of a slice of a brain. So this is what you are seeing here. So this is actually a slice of the mouse brain, and each of these dot in the center actually refers to one neuron cells. And you can see that they have all sorts of different colors, slightly different from each other, but never the same. In fact, in this study, they can achieve up to 90 colors just using combinatorial expression of fluorescent proteins in different cells. And this, of course, is a remarkable achievement in neuroscience, as well as in just technical development, because now with that, we can actually sort of distinguish every single cell that are in contact with each other inside a piece of the brain, which is really remarkable in my opinion. So, okay, so that's the second example about the fluorophores. The very last example I want to show you is takes a slight twist, because we are now going to the more physical side, which is something called super resolution microscopy. So what is super resolution microscopy? Well, as the name suggests, if we have super resolution microscopy, we should also have normal resolution microscopy, right? So what is normal resolution microscopy? Well, this is actually based on a key concept in optics, in the sense that the resolution of conventional normal light microscope, such as the ones that you probably have played with in your school, the resolution of that is actually limited. And this limit is something called a diffraction limit. Okay, and the exact physical basis behind that is quite beyond this talk. But for now, it's suffice to know that when light passes through a microscope, it can only be focused onto a finite sized spot, which is shown here in both X, Z, as well as XY. Okay, and this spot is basically called the diffraction limit, and it is basically the smallest possible size that any object that we put under the microscope will show up as. How small did it get? Well, in the horizontal dimension, it's about 200 to 300 nanometers, and in the vertical dimension, it's about twice of that. So what that means is that no matter how small your object is, as long as you put it under a normal microscope, the smallest possible size is going to show up as is this. It cannot be smaller than that. Okay, and this is a consequence of that is that if we have a structure that are smaller than this size, then we cannot see it clearly. And this is regardless of what brand of microscope we are using, whether we are talking about this 17th century old version that was used by Robert Hooke, or the kind of sophisticated equipment we can now buy for $500,000. Okay, it's regardless of what brand or model microscope you use. If your structure is smaller than the diffraction limit, what you're going to see is this kind of fuzzy, blurry images like this. Okay, so this is normal resolution microscopy. And what's the consequence of this resolution limit for biology? Well, we can get a better sense of that if we compare the wavelength spectrum of electromagnetic radiation, of which the visible light is just a small part. I'm sure you have seen that before in your school, right? So the wavelengths of the visible light is a narrow window from 400 to 750 nanometers or so. And if we superimpose this spectrum onto the spectrum of length scale that are relevant to biology, starting from the size of a human being, which is about 1 meter, then we can go down to the micrometer range, which we have all the kind of different cells, different eggs. Then we can go to the micrometer range, which are all basically we enter the cellular regime. And below 1 micron, where we enter the nanometer range, and that's actually where the really interesting stuff to biologists take place, or the intracellular structures and organelles and proteins. So as you can see, that the visible spectrum falls squarely into this range. And the diffraction limit, which is indicated by this blue line, is slightly lower than the visible spectrum. So what that means is everything to the left of this length scale is below the diffraction limit, which we cannot see clearly. And that is very, very bad for biologists because this is precisely the regime that the biologists are interested in. And this is precisely the reason why we want to break the diffraction limit to achieve super resolution microscopy. Basically, in other words, we want to turn this kind of fuzzy image into this kind of crisp, sharp images. Okay, and achieving this actually has been one of the major focus for the bioimaging community for the past 20 years or so, which culminated in the awarding of Nobel Prize in 2014 to these three scientists. And there are many methods that have been developed, but I just want to show you one of them, which is called the STORM, which stands for Stochastic Optical Reconstruction Microscopy. And so how does it work? Well, we can break it down molecule by molecule. So remember just now we said that in order to image biological samples, we need to first label them with some dyes of fluorescent proteins, right? In this case, let's say we want to label this particular fuzzy structure. And we can, of course, a standard label looks about this size. And we can do that for across the entire spectrum, entire sample by labeling many probes, right? But as you can see that the label is going to be very dense, which means that they tend to overlap with each other. So much so that when we take an image of the sample, we cannot tell them apart. And that, of course, leads to this kind of blurry image. So we can ask ourselves, instead of having them all emit fluorescence at the same time, can we somehow only have a small fraction of them emit fluorescence at each point in time? So much so that when that happens, they will be much more sparsely distributed, and therefore we can tell individual molecules apart from each other and not having them overlapped. And this principle is actually very nicely illustrated in this animation that I stole from YouTube again. So I'm just gonna let it run. So suppose instead of cell, we want to take a picture of the Eiffel Tower in the dark. How can we do that? What we could do is we can decorate the surface of the tower with lights, and then we switch on a few lights at a time. And we mark the position of these lights, and then we switch them off. And then we switch on another different set. And we mark their position and we switch them off again. So we can keep doing this until we record down the position of every single light on the surface of the Eiffel Tower. Then we can put all these positions together, and then we have a much better resolution image of the tower as compared to previously. So this, in the gist, is what super resolution microscopy is. Okay, so this is basically a repeat of the video I showed in the previous slide. So because of time constraints, I'm just going to skip through this slide. So basically, the principle of STORM is very well illustrated in the previous animation. So I just want to point out as the final slide on this super resolution microscopy, the physical chemical foundations of that, because as fancy as it may seem, the STORM or the other types of super resolution microscopy are actually all fundamentally rooted in physical chemistry. Why do I say that? For two reasons. Number one, our ability to very precisely identify the position of each of these individual molecules is the key to doing STORM, as you probably have already figured out. And that is actually doable because even though the image of a single molecule might be diffraction limited, but we can actually pinpoint the center position of it with much higher precision, down to an order of a few nanometers. Okay, and exactly how we can do that is not quite important. But let me just give an analogy. Okay, if you look at this mountain, I think everybody will agree that even though this mountain is very wide in space, right, we can still pinpoint the position of its peak very accurately. And that's, in some sense, the intensity of a single molecule image is very much like this mountain. It might spread across a very wide area, which makes it diffraction limited. But if we want to pinpoint the center position of this molecule, we can do that very, very accurately. So that is the first prerequisite for doing STORM. And which, yeah, in a different way, the precision with which we can localize molecules is much better than the resolution of the image. But that's only the first criteria. The second, in my opinion, even more important requirement is our ability to somehow control the switching, photo switching of these molecules, because that's what we want to do. We want to somehow control and fine-tune the density of these molecules that are detected at any point in time, so that we can ensure that the molecules are sparsely distributed, and therefore they can be accurately, each of them can be accurately identified. So there are various ways of doing various different types of the kind of photo switching behavior, some of which just called photo switching, which is a reversible switching between on and off state. Some of it is called photo activation, which is sort of the irreversible way we can switch on these molecules. And I won't go into the details of that. But you may ask how we can actually achieve this kind of photo switching behavior that actually can be done either optically, for example, by shining UV light on the molecules, or it can also be done chemically, for example, by carrying on redox reactions, and that converts the molecule between different states. So these are the three examples that I want to show you today. And because of running out of time, I will skip this slide about, which is sort of give a bit more bird's eye overview of what my lab is working on, which is divided into three main areas in terms of the kind of imaging methodologies we develop in terms of the biological system we applied it to, and also the physical chemical basis underlie all these various behaviors. But I think I'll just skip that. So my final slide, I just want to summarize and give you guys a couple of take-home messages, which I hope I have convinced you in the past 25 minutes or so, in that seeing is truly believing. And this is especially true when it comes to biological systems where really complicated things take place. And I've mainly focused on the optical approaches to doing the seeing part, particularly if fluorescence, which is the most popular way of doing visualization in biology. I hope I've also convinced you that biology over the past few decades has really become a quantitative science. And as such, it is imperative that we equip ourselves with a good understanding of the physical-chemical principles behind these life processes. Also, I've tried to demonstrate to you that the living cell nowadays, with all these technical advancements, has really become the new test tube for biochemists of the 21st century. And finally, because of such interplay of technologies and fundamental science, exciting new opportunities lie at the interface between chemistry, physics, and biology. And it is at this intersection that you guys will make a difference in advancing the frontiers of science technology with chemistry. And on that note, I'll end my talk, and I'm happy.
Now it's time to welcome our young scientist, Dr. Emit Lee Howe, to highlight on the topic chemical microbiology to study enzyme of the human microbiome. Welcome, Prof. Lee Howe.
Thanks, Radha, for the introduction. So let me just share screen here. Okay, I hope everyone can see my screen and can hear me all right.
Yes.
So good morning to everyone, especially the students for coming on Saturday morning. And so today I'd just like to share with you some of maybe a little bit of background on the human microbiome and how we use chemical tools to study the enzymes of the human microbiome. So I'll just start with a little bit of introduction. I think Radha has already done some of that. Just add on a little bit, I graduated from Hwa Chong, and I went on to do my undergraduate studies at University of Wisconsin and Madison in biochemistry. And during my undergrad studies, I became very interested in organic synthesis. So after I came back from my undergrad studies, I did a 1 year of research assistant in the lab of Professor David Chen and Casey Nicolau, whom actually MJ mentioned previously in his talk. So that's where I learned how to do a lot of the organic chemistry, organic synthesis, small molecule synthesis. And then I went on to do my PhD at Stanford, and I chose to do it in chemical and systems biology because I have very strong interest in both chemistry and biology, and especially the use of chemical tools for biological perturbations. And I came back to Singapore and did my postdoc studies in molecular engineering lab at A*STAR that was headed by the late Nobel laureate, Professor Sidney Brenner. And I just started out at NUS last year in August. And so this is our new S9 building where me and Minju are. So I'm on the 12th floor and he's on the 14th, right, I think. So please do come and visit. It's a pretty nice building and there's a Starbucks on the first floor. So, okay, I'll start by just a very short introduction on what chemical biology is. So this is the area that I work on, and I think some people might not be familiar with this area. So it is a very interdisciplinary focus where we really look at the study of chemicals and chemical reactions involving biological processes. And it involves the more traditional discipline of bioorganic chemistry, biochemistry, cell biology, and pharmacology, just to name some of the key fields here. And so what are some examples of chemical biology? Because it is actually quite a wide field. So I think one of the most defining areas of chemical biology, and probably how this term was initially coined, is the use of small molecule probes and inhibitors for biological perturbations. So this can be drug-like molecules, or it can just be any sort of inhibitors of enzyme reactions, and some probes that I'll mention later on. So beyond that, chemical biology also involves the development of new bioanalytical methodologies. And so one of the main highlights, I would say, is the advances in mass spectrometry, for example, and also next generation sequencing, and also what Winston just mentioned in his talk, all these new developments in better imaging techniques that allows us to really decipher what is happening at the molecular level. And another area of chemical biology that people actively work on is the evolution of proteins and biological catalysts. So this is in contrast to what I think Minju mentioned previously, that is on, you can think of it as non-biological catalysts. But in this case, we are talking about, for example, enzymes that you can evolve through multiple generations in the laboratory to form better catalysts. So what area do I apply chemical biology on? So for me, it is on human microbiome. So human microbiome is the collection of bacteria, fungi, and viruses in and on the human body. So the human microbial system that I think most of you, if you do know about human microbiome, would be our gut microbes. And so this is indeed where most of the human microbiome resides, especially in your large intestines. I think that's probably not surprising to most people. And but beyond the gut microbiome, there are also several important microbial communities, and that is the oral microbiome, the urogenital tract microbiome, and what I study quite a lot in my group, which is the skin microbiome. So what is it about microbes that are so interesting, and why do I want to study them? So microbes, in short, can expand our metabolic capacity. So what does that mean? It means that microbes can perform biochemical transformations that our body cannot. And this is because, we are coded by one genome, right? But we have lots and lots of microbes in us and lots of different genomes. So each of these genomes, they can have the capability to code for enzymes in particular that can perform these biochemical transformations. So the idea here is that because of these microbes in us, we are not just, you cannot just consider our body as what human genes are, but you have to consider it with the consideration that there's all these microbes that are also expressing all these enzymes and their associated products. So in this sense, the microbiome increases the biochemical transformation that our body can do. And so we want to understand these processes, and the way we do that is by really looking at a key component, which are the enzymes that produces these molecules. But we also want to look at it in the context of the host and their interactions with the microbes. So in short, our goal is to really look at, in a molecular level, how these microbial enzymes and their associated products, their substrate, how this will structure the human environment, both in health and in disease. And so some of the techniques that we use in the lab involves microbiology, both traditional microbial cultures where I grow up the microbes, but also culture-independent microbiology, which is where you directly sample from various sites on the human body, and you can assay for the community makeup of the microbes, or you can also look at various enzyme activity. We also combine functional genomics and advanced tools in molecular biology to look at the expression of certain enzymes. And more importantly, we incorporate chemical tools to study a lot of these microbes. So we use inhibitors and probes, and also mass spectrometry to look at what are some of the molecules that is being produced by these microbial enzymes. So I'm just going to introduce one topic in our lab. Oh yeah, so I should mention also that we also go into mouse models and organoids to look at what are the effect of these molecules. So one topic that we focus on is the enzyme mechanism-based profiling of the human microbiota. And in this project, what we do is that we first make or design tools that allow us to look at the enzymes that are produced by these human-associated microbes. So this involves chemical tools such as enzyme substrates and activity-based probes, which I will mention later on, and also molecular biology tool, which I'll mention in the next slide. But overall, these tools will allow us to detect what are the changes in the enzyme expression and activity when we compare the microbiome community in a healthy population versus the disease population. And so using these tools, we want to identify enzymes of interest. And from this, we can study these enzymes more in detail by, for example, making these enzymes in the laboratory. Can also perform structural studies to look more into the enzyme catalysis, and also their effect on the host, in particular human, by doing in vitro human models. In this case, what you're seeing here is a 3D skin model that we do in our lab. So just to go further into two of these tools, so now I'm going to talk about how we look at the RNA that is expressed by microbes. So how do we assess this RNA that's expressed by the microbes? So in case those of you who are not familiar, RNA are transcribed from DNA. So, they are basically, we look at the messenger RNA, which then is eventually get translated to proteins. And we look at the RNA to give us basically an idea of how much of each of the genes are being expressed. And how do we do that in a non-invasive manner? Because we're not going to find many volunteers if every time we want to do this sampling and I need to do a small skin surgery. So what we do instead is we use a non-invasive technique that is what we call tape strip. And this is really just an adhesive circular disc, and it's pretty much like a Scotch tape. And what it does is that just by sticking it onto your skin multiple times, we're able to sample the very top layer of your skin, which is known as the stratum corneum. And I think some of you will know it as like dead skin. And so this technique can allow us to actually very cleanly sample the top layer of the skin. And we find that when we isolate the total amount of RNA, that it is actually a tiny, tiny amount of RNA that we isolate. So with this tiny amount of RNA, how do you look at its expression? So the way we do it is using next generation sequencing. And so this is a little bit complicated, but in short, what it means is that when we have the RNA, what we can do is to first reverse transcribe it into DNA. And once you do that, that allows us to do very robust sequencing on the Illumina platform. And so what the results look like. So what this graph shows you, it is a principal component analysis, and that is a statistical analysis method to look at multiple variables in a 2D manner. So for example, you can have, in this case, we have an expression of about 50 enzymes. In this principal component analysis, what you can see essentially is that there's this separation of the purple and the blue dots with the green dots. And what the green dots are the samples that we derive from the healthy volunteers, and the purple and blue dots are from patients with atopic dermatitis or seborrheic dermatitis, which are two skin conditions that is quite common in Singapore. So atopic dermatitis is, I think, commonly known as eczema, and seborrheic dermatitis is a more severe condition of dandruff, where we see flaking both on your scalp and also on your face. So from here, what we can essentially identify is differences in expression between different enzymes. And from here, we can follow up on what are the interesting candidates that might be involved in these diseases. So this is looking at the enzyme expression used at the mRNA level using molecular biology tools. So another tool that we use very commonly in the lab is activity-based probe. So what are activity-based probe? So I think to link this back to Winston's talk just now, so he mentioned that to visualize a lot of the proteins in a cell, you can attach a fluorophore. And so how do you attach a fluorophore? In some cases, especially if the protein is an enzyme, you can use a chemical attachment and to basically covalently link a fluorophore onto the protein of interest. And the reason why I say that it's works mostly only for enzymes is because this technique requires the covalent reaction between the nucleophile, which is in the active site, with the electrophile on this activity-based probes. So some example of activity-based probe structures are shown here. And you can see that they generally consist of three main parts. So first is the electrophile that I've mentioned previously, that you can reacts covalently and irreversibly with the nucleophile or in the enzyme active site. For example, here you have the vinyl sulfone or the epoxy ketone as the electrophile. And you have affinity scaffold, which will direct you to the enzyme of interest. In this case, these probes are against proteases. And so the affinity scaffold are peptide sequences that are specific to the protease of interest. And then finally, you can attach a fluorophore here, and this allows you to visualize the protein. What is also commonly done is instead of fluorophore, we can attach what is called an affinity handle. And what an affinity handle is that it can allow you to enrich the protein out of a complex mixture. So again, how these probes work is that when you incubate the probe with their enzyme of interest, this probe will get into the active site of the enzyme. And because this is a covalent reaction, you can, for example, when you run a protein gel, you see that only enzymes that are labeled by the activity-based probe will light up. So in this case, for example, this vinyl sulfone probe, it will react in a irreversible manner with the N-terminal threonine, which is the nucleophile in this enzyme, and form you can see this covalent product. So what do we want to use this activity-based probes for? So there are many ways, there are many functions. So one I've mentioned is you can use it for imaging, and another is that you can use it for new enzyme discovery. And for us, what we're interested in, again, to bring back to the idea of human microbiome, is that we want to use these tools that I have mentioned to identify secreted microbial enzymes that can be biomarkers in disease. And we want to basically look at the difference in activity, difference in expression, to see if any of these microbial enzymes can be a prediction of clinical disposition for certain skin disorders and also the outcome. And what we do is that we collaborate quite extensively with clinicians in Singapore. So here are some of the skin conditions that we study. I've mentioned atopic dermatitis and also psoriasis in collaboration with National Skin Center. Psoriasis versicolor, which is actually a very common skin condition in Singapore, as well in hot and humid countries, is this sort of whitish, scaly rash that appears and is non-itchy. Apparently very common with in army boys. And so we had this collaboration with NUH dermatology, and also diabetic foot ulcers with St. Luke's Hospital. So I hope that from my talk that you can see how we use chemistry to really look at different mechanisms in biology, and also how we can use chemical tools to, for example, identify biomarkers, and that can help us to advance medicine. And so I think this highlights that chemistry is what I think indeed what many people call central science, and can help us to really understand many different areas. So that's all I have.